All right, for our third speaker this morning, we have Associate Professor Arthur Gonzales. The third, he finished his bachelor's and master's degrees in chemical engineering at the University of the Philippines in Diliman. He finished his PhD in Chem Eng also at Northeastern University in Boston in the US. Currently, he's an associate professor of the Department of Chemical Engineering and deputy director for the human resources of the university. His research interests are into multi-scale molecular modeling, molecular dynamics, molecular docking, among others. So to give a talk on computational testing of phytochemicals for the inhibition of the main protease of SARS-CoV-2, let's give a warm welcome to Professor Arthur Gonzales. Sir Bok, go ahead.
Right. Thank you, Joey, for that introduction. Let me just play my slide. All right. So welcome, everyone. I'm happy to be able to talk to you about our work in the identification of inhibitors against SARS-CoV-2's main protease using virtual screening. This has been a project or initiative by the UPD College of Engineering COVID-19 response team. And I'm working on this with Dr. Remolona, who will present about his work later. So we all know what causes COVID-19. It's the SARS-CoV-2 virus. It is, as noted by the New York Times, a bad news wrapped in protein. It has infected more than 15 million and killed over 600,000 people worldwide. And it is a single-strand RNA virus wrapped in a lipid bilayer with structural proteins that we know binds very strongly or efficiently with its host. It's a new virus, therefore we don't have any antibodies against it. That is why we think that the best approach to conquering this pandemic is drug repurposing. Novel drug molecules take a long time to develop. So this is the usual drug development pipeline. So from the discovery part, preclinical, 3 phases of clinical trials, and then the long winding road of approval. And safety trials, of course, and regulatory barriers cannot be bypassed for new drugs. Immediate solution with known safety, availability, and affordability is needed. So we all know what happens when we sidestep all these regulatory barriers. The solution becomes deadlier than the disease. So we think that drug repurposing is very apt because approval for repurposing could be more quickly achieved with emergency provisions such as a response to a global pandemic. So our focus of our study for now is a study of plants phytochemicals in the Philippines for its potential medicinal purposes. So it has been noted that preclinical, clinical, and epidemiological research suggests that phytochemicals can indeed be effective in treating various diseases owing to their antioxidant and anti-inflammatory activities, as we have seen. And the Philippines, our country, is really a powerhouse in terms of phytochemicals. We are one of the 17 megadiverse countries. In fact, we have 80% of the known species in the world, with about 16,000 plant species, where it has been estimated that 45 to 60% of that 16,000 is endemic. So the focus on the phytochemicals is in consideration for the mass production of such a therapy in case of verification. But to start this drug repurposing, we have had these challenges. So at the beginning, around January or even in December, we lacked molecular information about the virus. If we have some information, we lacked or we were wanting for biological data. Initially, there was an incomplete genome sequence, and because of that, then it resulted to a non-availability of the crystal structures of the target proteins. And we have a small library of compounds. Small meaning these compounds were from those studied for SARS, the original SARS, and MERS. But after a couple of months, all of these have been solved. So we have this figured out. In the Philippines, though, I think the challenges are that we have limited computing resources for in silico and in vitro and vivo studies. Limited accessibility to access the efficacy of identified leads in vivo and in vitro, as I mentioned. And the question of, can we have clinical trials here in the Philippines? And I think with concerted effort by the DOST, UP, UST, and several other academic and government institutions, I think I can say that we have also resolved these challenges. It's not comparable to our international counterparts, but I think we have enough to start. And so with that, let me show you a brief structure of the methodology, which is the structure-based drug discovery and design. So our tools are molecular modeling methods. And we aim to for the first part, we have to cast the net broadly to expand the search space. This is where Professor Remolona will come in later, actually. We also have to increase the chemical diversity of the leads. So if they are very similar, if one fails, then almost everyone will fail. So we have to increase the diversity. This will also give us more tight binders and tighter binding. So more binders and larger binding energy. And we need to optimize the potency of leads, and we can do that with this. And this could also potentially reduce off-target effects by, so the power of molecular modeling and structure-based drug discovery is the exhaustiveness and the scale. So we can increase the scale. So we can potentially reduce off-target effects using those. And this could be used to mitigate attrition rate in preclinical and clinical studies where about 90 to 95% fail in the clinical trials. And finally, the main goal, I think, of this method is to generate a buy list, meaning we have the potential compounds, we buy them, and we test them in vitro and in vivo. So several strategies have been used by many laboratories, and I think you're familiar with this. So the several target proteins, I'll name 3 here. So we have the spike protein. This is the one that binds with the ACE2 very, very efficiently. That's why we are prime candidates for this kind of virus. We have a lot of ACE2 receptors in our bodies. So a target of a lot of studies around the world. Vaccines, cures also are targeting this target protein. So it's tricky to hit because of changing conformational states, as you can see here. It has an open state and a closed state. And by the time I think around March, they're not clear. It was not clear yet what parameters can make the spike protein open up. But the ACE2 inhibition has been explored more widely, so we have a starting point for this type of strategy. The other popular one is the polymerase. So this guy stitches and corrects RNA strand to create new virus particles. It is a big target protein, so there's a disadvantage of doing this in molecular modeling, because it's a very large protein. So it also has a few conformational states, which again makes it difficult to target. And it is a target of a lot of candidate drugs already approved for treatment or even just for clinical trials, for example, Remdesivir. So our lab chose this guy, the 3-cysteine-like protease, the main protease. It cleaves the polypeptides at, I think, 11 sites, no? To produce different enzymes for virus reproduction. It is an attractive target because of its known stability and known active site, which is this part actually. And it is very similar to other coronaviruses proteases. So as I mentioned, so it has already been done. The compound libraries used for SARS and MERS, those were the initial test compounds to tackle this protease. And tackling the protease actually gives us a potential solution to many infections and future problems, as I will note a bit later. So as a summary of the methods, we use the protease, as I mentioned, and that's our target protein. We prepared that. So what's the pH? What's the ionization state? And then we also did MD simulations on the protein to give us 5 representative structures. And then we prepared the target or the ligands. Actually, this started with around 2,000. And then we had a initial screening, so Lipinski's rule of 5 for pharmaceuticals, and we have eliminated any toxic phytochemicals. So we started now, or so we have now 1,103 phytochemicals from various databases. And then we generated the ionized states, resulting to 1,800 unique compounds. And then we docked the compounds to rank them using Schrodinger. And then we identified the top 16. Actually, we have more than that. And then we did MV simulations to analyze stability and protein-ligand interactions. We also did the procedure for 14 reference compounds. So these are compounds such as Remdesivir, which has been tested to be, I guess, effective against SARS. So we have 14 control compounds. And then we did a very exhaustive absorption, distribution, metabolism, excretion, and toxicity in silico evaluation. So here are the results. These are our top 16. These are the structures of the top 16. This is a snapshot of one of the top candidates. I think that's compound 0863-1. This is a docking pose. We can also identify the active residues interacting with the candidate. So these are those. What's interesting to note here is that the histidine 41 and where is that? So yeah, histidine 41 is one of the catalytic, cysteine 145, there you go, is that pair is the catalytic dyad, and they are interacting with all of these. They have interactions with all of these. After MD simulations, we plotted the root mean square deviation, and all of them seems to have or indicate a stable complex after around 5 nanoseconds. So after 5 seems to be stable. And visual inspection told us that no candidate exited the binding pocket. We also evaluated the root mean square fluctuation that will give us the active residues. This is an example of that for, again, 0863. And we can also note that all candidates have interactions with the catalytic diet histidine 41 and cysteine 145. We also did time-dependent interactions analysis. For the top performer in the MD simulation, we have the aurantiamide acetate here. And we can analyze, after analysis, we can see here that histidine 41 has different types of interactions with the protein. So it has H bonding, hydrophobic interaction, and water bridge interaction. Here's a summary of the results. So we have 16. We have glide scores between 8 and 9.5, and a binding energy between 43 and 90. We also did a, as I mentioned, exhaustive ADMET analysis. The summary of that can be seen here with the quick prop score. So we used more than 40 molecular descriptors. So the highest performers are 096, 537, 1206, and 1201. We also analyzed several drug-like properties such as QP log, polar surface area. We also noted the Lipinski violations, rule of 5 violations, and finally the percent human oral absorption if we are going to make this drug, taking this drug orally. So we can see 096 and 1206 have very high oral absorption at 195.3. This is the summary of our test compounds, or sorry, the reference. The binding scores are very similar to the test compounds. Let me point out here that Remdesivir also scored high at -8 kilocalories per mole, and this is against the 3CL Pro. So remember, Remdesivir was designed to interact with the polymerase. So this tells us that it might also be interacting with the protease. So in conclusion, we use structure-based drug discovery methods, molecular modeling methods, to identify potential inhibitors against 3CL Pro. These compounds, the top 5 compounds of the 16 actually, are arantiamide acetate, isoliquiritigenin, luteone, and apigenin. These are the sources, plant sources. And we recommend these 5 compounds to be tested in vitro and in vivo. In fact, we are in communication with the biochemistry department of UP Manila for potential collaboration in testing these compounds in vitro and in vivo. We're just waiting for some funding. The results will be used in machine learning experiments by Professor Rimalona to potentially accelerate the screening. So as I mentioned, we screened 2002 compounds initially. With machine learning, we can potentially expand that space to hundreds of thousands or even millions of compounds. But as a note, we have to understand our limitations as well. So the values that I gave you guys do not represent the actual binding energies. but they are, they are close. And they only provide snapshots of interactions, even with the molecular modeling interactions. And a warning that 95 to 98% of repurposing fails, but we need just one or two to work, and then we should be fine. And in the Philippines, we are behind in funding and infrastructure to perform researches like these, but we have the talent, from the modeler to the chemist to the biochemist to the clinician. We have the talent, but we somehow lack funding and infrastructure. I want to leave a few notes. Is that this is not the first nor the last pandemic that we are going to face. Initially SARS, and then MERS, and now SARS-CoV-2 or COVID-19 all caught us unprepared. There was some pharmaceutical efforts, especially when SARS emerged and MERS emerged, but then they were quickly dropped because the disease just disappeared. Why? Because the incentive to risk ratio to develop vaccines and cures for future diseases such as SARS, similar to SARS-CoV-2 or COVID-19, may not be high enough for many pharmaceutical companies. That is why the research should be initiated by government institutions and the academia with help from some angel investors. And the program should have an industry partner to quickly test, manufacture successful candidates. And I think for this, there are two ways to success: repurposing of available drugs and phytochemicals. And after we have succeeded against SARS-CoV-2, I think we can design new drugs to help prevent this kind of pandemic in the future. I'd like to thank the College of Engineering, OVCRD, DOST-RDT, and UP Engineering Research Development Foundation Incorporated for the funding for this research. And thank you guys for listening. I'll be happy to answer any of your questions.
Thank you, Dr. Gonzales, for that timely talk on drug discovery using molecular modeling and molecular simulations. We're now